Well, good afternoon, everybody. Welcome to day 2 of the first virtual internal medicine research days now. We appreciate you for taking the time and joining us. For today's keynote speaker today, we're privileged to have with us, albeit virtually, an outstanding clinician scientist, Dr. Joseph Wu. Trust me, I too need a dual monitor setup. I need this separate, very large monitor to serve as the summary and reference of all of Dr. Wu's accomplishments and accolades through his career. Dr. Wu received his medical degree at Yale University, after which he completed his residency and fellowship, as well as his PhD training, all at UCLA. Dr. Wu is currently a professor and director of Stanford Cardiovascular Institute, where he conducts research implementing multi-omics approaches in the realm of cardiovascular medicine. He's truly a pioneer in the field of cardiac stem cell biology. Particularly, he's really using cardiomyocytes derived from patient stem cells as a means to model disease on a personal level and to serve as a platform for drug discovery. In doing so, he's really pushing the field forward, particularly in this era of pharmacogenomics, personalized and precision medicine. Dr. Wu's research has always been of high impact. He has an H-index of 100 with over 400 publications, and as a result has earned a number of high-level awards, including a Presidential Early Career Award, one that was handed to him personally by President Obama. And also from the American Heart Association, he's received both a Merit Award and a Distinguished Scientist Award. Beyond these, he has an exhaustingly long list of R1 level NIH grants that are currently active. If I counted correctly, it was some mind-blowing number, like 13 or so. Dr. Wu also serves on several editorial boards of high-impact journals, for example, JCI and Circ Research. And he's also chair of the American Heart Association's research committee and also serves on their national advisory board. As one might expect, he's also an elected member of several prestigious academies and associations, including the American Association of Advancement of Science and the National Academy of Medicine. In addition to being a well-respected and highly prolific clinician scientist, Dr. Wu is deeply committed to training the next generation of physicians and scientists. And this is really exemplified by the fact that 30 of his former postdoctoral trainees now have faculty positions of their own. It's quite impressive. Dr. Wu is always very kind and has been very approachable. I myself have interacted with him at several conferences and have emailed several requests for either shipment of some of these iPS cardiomyocytes or seeking information on one of my AHA grant submissions. I imagine he gets plenty of these emails of this nature, and each time he's been so responsive, and I think it's just a true highlight to his commitment to serving the much broader research community well beyond his own lab. So I'm pleased to thank Joe for joining us today, and I'm excited to hear his keynote seminar entitled Stem Cells and Genomics for Precision Medicine. Thank you, Joe.
So thank you, Ryan. Thank you. It's a tremendous honor for me to speak at the Internal Medicine Research Day and to see many of my friends at Yale on the Zoom webinar, many others. And so, I'm very grateful for this opportunity. And so I'm going to talk about, I'm a cardiologist, and so I'm going to focus on how we use stem cells and genomics for precision cardiovascular medicine. And before I start, this is my disclosure statement here. I think, as many of you know, there's a significant interest in precision medicine or precision health in the US. This concept was promoted by President Obama back in 2015. And much of this is made possible by recent advances in next generation sequencing, bioinformatics, and big data. the concept is that by understanding our genes along with lifestyle and environment, we can then better predict which treatments might work best for us. Now, in the case of DNA sequencing, the technology has now become much cheaper with lower error rate, broader coverage, and much faster turnaround time. So back in 2000, it cost about $1 billion and over 13 years to get your DNA sequence. Today, you can get your DNA sequence for less than $1,000 in 2 weeks. With increasing usage of next generation sequencing, there's hope that care can be more predictive, preventive, personalized, and participatory, the 4 Ps of precision medicine. However, for many clinicians, including myself, I've always wondered what exactly is precision medicine. Now, while there are many definitions, including finding the best treatment for the right patient, I would like to paraphrase Professor Dan Ariely from Duke University here. So back in 2013, when he was asked about big data, he famously tweeted, I won't read the first sentence here, but I'll read, everyone talks about it. Nobody really knows how to do it. Everyone thinks everyone else is doing, so everyone claims they're doing it. So to a certain extent, it's the same for precision medicine because oftentimes it's a buzzword in healthcare used by all the hospitals for advertisement. Now, to be more scientifically correct than Professor Ariely's tweet, I think the major goal of precision medicine is that with the help of next generation sequencing, we can pinpoint a patient's genetic risk of developing, in this case, cardiovascular disease, and then use their genetic information to find the optimal treatment for individual patient. However, just like anything else in medicine, the reality is that it's much, much, much more complicated. Now, for starters, there are hundreds of genetic risk variants with small effects for most common diseases. And thus, it is very difficult to develop a clear picture of who is really at risk and for what. Now, one reason is that besides DNA, there's a complex world of epigenomics, transcriptomics, metabolomics, and environmental omics that are not captured by DNA sequencing. Another way to say it is that the cells in our body has the same DNA sequence, yet what is being transcribed and translated in each cell type is very different, which is the reason why we have different tissues and organs in our body like the brain, lung, liver, kidney, and heart. Now, ideally, we would like to study patients' heart tissues directly so we can accurately measure tissue-specific changes in response to drug treatment. For example, if I take a statin or ACE inhibitor or beta blocker, what exactly does it do to my heart muscle cells or blood vessels, besides just lowering my cholesterol and blood pressure? But that's not possible because we do not have easy access to human heart tissues or blood vessels, and hence we typically use blood as indirect biomarkers for many of the drugs that we take. For example, we do microarrays or RNA sequencing of the PBMC to give us a hint of what's going on for Alzheimer's disease, for heart disease, which to me absolutely makes no sense because PBMC are not the same as brain cells or heart cells. Now, this has now changed with the human iPS model. Now, keep in mind that these human iPS cells are still not the same as the human heart, but it's as good as it gets in terms of our current technology in 2020. iPSL is a powerful platform because it is genetically identical to the patient, and it encapsulates the patient's DNA, RNA, and proteins to give you a phenotypic readout. So for example, if you have polygenic risk variants, instead of trying to guess the functionality of each variant, the iPS cells may provide you with a cellular platform to understand the composite phenotype of all these genetic variants. So, as many of you know, this technology was first pioneered by Dr. Shinya Yamanaka around 2006. The discoveries led to him receiving the Nobel Prize in Medicine Physiology in 2012. process involves taking patients' skin or blood, transfecting the somatic cells with reprogramming factors such as OCT4, SOX2, KLF, and c-MYC to derive the almost the equivalent of human embryonic stem cells that can both self-renew and are pluripotent. And just like these human ES cells, these iPS cells can then be differentiated into any somatic cell types, such as cardiomyocytes, neural cells, or islet cells. This then allows you to characterize the disease phenotype, identify molecular targets, and implement personalized medicine. So currently, our lab has generated iPS cells from more than 1,000 patients with different genetic background and cardiovascular diseases. From these diverse population of patients, we can then generate, for example, 2D micropattern human cardiomyocytes, 3D human cardiac organoids, and 3D engineered heart tissues, as shown here. Perhaps the most important advantage is that these come from human samples, and hence it can be used to recapitulate the diverse population of human genetics that is otherwise not possible with traditional animal models. And so for the next 30 minutes, I'd like to show you applications in 3 major areas. The first area is elucidating cardiovascular disease mechanism. The second area is using iPS cells for cardio oncology. And then the third area is the concept of a clinical trial in a dish. So shown here is a schematic of different causes of heart diseases, which falls into 3 major buckets. The first is primary cardiomyopathies, which could be genetic or acquired. Genetic cardiomyopathies would include, for example, hypertrophic cardiomyopathy, dilated cardiomyopathy, restrictive cardiomyopathy, left ventricular compaction, and ARVC. Acquired cardiomyopathies would include viral myocarditis, which is quite timely given the current COVID-19 pandemic and reports of COVID-19 related myocarditis. Next is secondary cardiomyopathies, which could include amyloidosis, iron overload, hemochromatosis, and chemotherapy-induced cardiotoxicity. And then the third bucket is the more common causes that are likely polygenic, such as coronary artery disease, hypertension, and congenital heart disease. And I will show you several examples of how we use iPS cells to understand the mechanisms of these diseases here. The first example is a familial dilated cardiomyopathy, or DCM, which is a frequent cause of ventricular tachycardia and VFib, requiring AICD implantation in patients. The prevalence is about 1 in 2,000 patients, although it is most likely underestimated. And the reason is that with increasing use of DNA sequencing, many patients who were initially diagnosed with non-ischemic or idiopathic cardiomyopathy are now found to have a strong genetic component. So about 10 years ago, we will refer this family by our colleagues at Stanford. The boy here was 14 years old when he had a heart transplant due to severe heart failure symptoms. Turns out his father, uncle, and grandmother all had dilated cardiomyopathy. Based on that, genetic testing for DCM panel was then ordered and identify a point mutation with arginine tryptophan switch at the amino acid position 173 in the cardiac troponin T protein. Now, besides giving them the standard treatment of ACE inhibitor and beta blocker and spironolactone at that time, the question that was asked by our referring physicians and patients themselves was, how does this troponin T mutation cause the severe clinical phenotype? So we decided to generate iPS cells from the entire family and differentiate them into cardiomyocytes, both affected and unaffected, so that we can compare their differences. And compared to the healthy controlled cardiomyocytes, the DCM cardiomyocytes show altered regulation of the calcium ion, decreased contractility, and abnormal distribution of the sarcomere alpha actinin. And importantly, treatment with the drug beta blocker, for example, metoprolol, improved function of the DCM cardiomyocytes, recapitulating results from multiple large beta blocker clinical trials. So this study was published 8 years ago in 2012. And you see subsequent samples from our lab and from other labs as well. Now, another example is familial hypertrophic cardiomyopathy, or HCM. HCM affects about 1 in 500 people. Clinically, these patients have left ventricular outflow tract obstruction and cardiac arrhythmia. HCM is one of the most common causes of sudden cardiac death in young adults. It is the diagnosis of exclusion when an athlete passes out in the football field or basketball court. And more than 100 mutations have been identified across 30 genes and so forth. So about 8 years ago, we were referred a large family with familial hypertrophic cardiomyopathy by our heart failure cardiologist. The mother has HCM with symptoms of cardiac arrhythmia. She has 8 kids. The 2 oldest ones already had HCM phenotype, whereas kid number 3 and kid number 8 are both genotype positive but phenotype negative, most likely because they were still young at that time. Through a whole series of experiments, we were able to show that this particular myosin heavy chain 7 mutation with arginine to histidine switch at codon 663 causes increased intracellular calcium, which leads to calcium handling defect and then arrhythmia. It also causes activation of the calcineurin NFAC signaling pathway, which leads to hypertrophic response. Importantly, treatment with verapamil and beta blocker can be used to reverse both cellular hypertrophy and cellular arrhythmia. So right now you can start seeing a trend of how we use these cells as an in vitro platform for drug screening. Another type of cardiomyopathy is ventricular non-compaction, or LVNC. LVNC accounts for 9% of children with primary cardiomyopathies and is the 3rd most prevalent form of cardiomyopathy after HCM and DCM that I just discussed. Now, it is characterized by hypertropregulation at the left ventricle due to arrested development of the left ventricular muscle during embryogenesis. Patients can present with either dilated or restricted physiology. Patients can also have severe arrhythmia that can lead to sudden cardiac death and oftentimes require AICD. Now, young patients who have never been diagnosed before sometimes present with stroke symptoms due to a blood clot that form in these spongy holes right here. So about 6 years ago, we will refer this large family cohort from Stanford Pediatric Cardiology Clinic. We also study a smaller family from Japan because the first author, Kazuki, was a postdoc from Japan. The boy here already had a heart transplant at Stanford. His older brother and older sister also had LVNC. Both of them were asymptomatic. Interestingly, his father had mild dilated cardiomyopathy without LVNC, and his paternal grandmother had cardiac arrhythmia requiring pacemaker. So through a series of experiments, we were able to confirm that mutations in cardiac transcription factor TBX20, one in the T-box domain for the Stanford boy, and the other in the transactivating domain for the Japanese girl, caused this abnormality, which capitulate a key aspect of the LVNC pathophysiology. A more recent study, we will refer to a family cohort with Lamin AC mutation by our electrophysiology colleagues. Now here, the presenting symptoms for the proband with severe arrhythmia requiring AICD and later heart transplant. And as you can see, this is a large family with affected members in multiple generations. Many of the individuals had cardiomyopathy, atrial fibrillation, and atrial ventricular block, and 8 of them actually died from sudden death. So it's quite devastating for the entire family with these terrible losses. So using iPSC-derived cardiomyocytes, we confirm that the Lamin mutation is indeed responsible for the cellular arrhythmia and impaired contractility. And as shown in this cartoon here, the typical control healthy cardiomyocytes have normal expression of lamin protein, lamin nucleus structure, and lamin associated domain. However, patients with lamin AC mutation have abnormal lamin nucleus structure that causes increased open chromatin in the lamin associated domain. This then leads to activation of genes that should have been silenced in otherwise healthy conditions. And using ATAC-seq and RNA-seq, we identified a PDGF signaling pathway as one of those abnormally activated genes in the mutant cardiomyocytes compared to the control cardiomyocytes. And importantly, we also showed that by blocking the PDGF pathway with FDA-approved drugs such as tyrosine kinase inhibitors, quinolinib and sunitinib, a very low dosage can actually be used to rescue the abnormal phenotype of these mutant cardiomyocytes. So now, so far, what I've shown you are considered classic Mendelian genetics with autosomal dominant or autosomal recessive inheritance, right? But human genetics, as I mentioned, is much more complicated, much more messy, especially dealing with variants of unknown or uncertain significance, which is really a clinical conundrum for clinicians taking care of these patients. So for example, shown here is the proband of a healthy middle-aged man who underwent DNA sequencing as a part of an executive VIP workup. He was found to have a likely pathogenic variant in myosin light chain 3 with cysteine to alanine switch at codon 170, along with several members of the entire family, including his mother, brother, and also his daughter. Now, I already showed you earlier that SCM can cause cardiac arrhythmia and sudden cardiac death. So what do you do in this case then, right? You leave the patient alone because he's currently asymptomatic and neither is his mother and brother. Now, if so, hypothetically, he flies from San Francisco to Australia on a business trip. It's a long 15-hour flight. He develops a blood clot in his leg, gets a pulmonary embolism, and then dies. During the autopsy, the doctors missed pulmonary embolism lodged in small pulmonary arterial branches. So in that case, what is the official death certificate most likely going to say? Well, unexplained sudden cardiac death. Now remember, he's a VIP patient, and even worse, his wife is actually an attorney. So what's going to happen next? Lawsuits, right? Now, as the concierge doctor who ordered this genetic testing for VIP workup, hypothetically, you decided to practice more of a defensive medicine. You then order a cardiac MRI to make sure there's no cardiac fibrosis. You order a 2-week Holter to make sure there's no cardiac arrhythmia. You also order an exercise treadmill to assess exercise-induced arrhythmia. All of them came back negative, which is quite reassuring to you. But in all these workups, you've kind of spooked the patient a little bit because next time when the patient is awake at 2 a.m. in the morning, working on the deadline, drinking too much coffee, feels slight palpitation, he's going to call you because he has 24/7 access to you as his concierge staff. So you can see that these scenarios that I just highlighted are not too far-fetched and will happen more and more as we start performing DNA sequencing on a whole bunch of healthy asymptomatic individuals. So this case was actually referred to us for further adjudication using iPS cells and genome editing. And so here we first generated iPS cells from family members and then showed that their cardiomyocytes were normal at baseline. Now, second, when we introduced a homozygous variant by CRISPR editing, meaning that now the cells has 2 copies of this particularly pathogenic variant, the cardiomyocytes also remain normal. Third, when we disrupted the healthy allele by introducing a frame shift mutation, the cardiomyocytes were also normal, meaning that what was annotated as potentially pathogenic variant allele was still able to perform normal function. And finally, just to make sure that our assay is working correctly, when we genome edited a well-known pathogenic variant of myosin light chain 3, his cardiomyocytes then show severe arrhythmia. And then based on these 4 steps, and based on the fact that the patients were clinically healthy, asymptomatic, we concluded that what was initially annotated as likely pathogenic variant is most likely a benign variant. Now, keep in mind that further studies would still be needed to validate this concept of combining iPS cells with genome editing for adjudication of variant of unknown significance. But I think if proven to be effective, this could have a significant impact on how we manage VUS patients in the clinic, especially when more people are getting their DNA sequenced these days. Now I want to shift gear to the second topic, which is using iPS cells for studying cardio-oncology. This is a major problem because heart disease and cancer account for the top 2 causes of morbidity and mortality in the US. As more cancer patients survive due to better chemotherapy and immunotherapy, such as checkpoint inhibitors, more patients are ending up with heart failure symptoms due to potential cardiac toxicity. So the question is, can we somehow use iPS cells as personalized therapy to assess chemotherapy-induced cardiac toxicity, as shown on the right here? Now, there are several logistical hurdles, including time, cost, maturation of the iPS cell cardiomyocytes, efficiency of scale, and so forth. However, at the same time, the process of making the cells is becoming cheaper and faster. Differentiating cardiomyocytes is becoming more reliable, and phenotypic readouts are also more robust than ever before. So I want to show you an example of how we apply iPS cells for cardio-oncology. Now, I must admit that one of the reasons why I study, in this case, breast cancer in particular, is that on my wife's side, there's a strong family history of breast cancer. So about 70 years ago, my wife, who's not a scientist, asked me to do something important rather than just playing around in the lab with the iPS cells. And that's the main reason why we decided to focus on breast cancer. And I think, as many of you know, breast cancer is the second leading cancer death in women, with more than 12 million cancer survivors in the US. 8 to 10% of them will experience doxorubicin-induced cardiac toxicity acutely. And among the survivors, 20% will develop heart failure over 5 years. 40% will develop heart failure combination of doxorubicin and Herceptin more than 75 years old. And so the current treatment guidelines giving patients beta blocker and ACE inhibitor once the echo shows reduced LVEF. And the question is this considered precision medicine as well? Now, showing the study we published about 4 years ago. We recruited 8 breast cancer patients, and a year later, some of them had normal cardiac function, while others have doxorubicin-induced cardiomyopathy. We generated patient-specific cardiomyocytes and exposed the doxorubicin in vitro. We then studied cellular responses by contractility, calcium cycling, metabolism, gene expression, apoptosis, and chemoprotection assays. And then we showed as proof of principle that if we pick the patients appropriately, iPS cells can be used to recapitulate which patients are more susceptible to doxorubicin-induced cardiomyopathy. So this slide shows the patient profiles. We recruited 4 healthy individuals as control. We recruited 4 breast cancer patients who were treated with doxorubicin and tolerated fine, which we call DOX group. We also recruited 4 breast cancer patients who were treated with doxorubicin and developed toxicity, which we call DOX-TOX group. Now, none of these patients had other cardiac conditions like previous myocardial infarctions or valvular diseases or heart surgeries before. So for example, here you have a relatively young 40-year-old woman whose baseline left ventricular ejection fraction was 77%, but then went down to 36 to 50% after 4 cycles of doxorubicin treatment. We exposed the cardiomyocytes and the DOX in the DOX-TOX patients to the same dose of doxorubicin. And then at 1 micromolar, you can see the DOX-TOX group had more sarcomere disarray, suggesting more damage to the cellular architecture. At 1 micromolar, the DOX-TOX cardiomyocytes had more cell death, more cellular arrhythmia, and more cellular apoptosis. So this is quite interesting here. Even though we do not know the exact variants or SNPs that could account for the patient-specific differences in chemotherapy-induced cardiotoxicity, the iPS cells is now serving almost like a mini chip readout that incorporates all the patient's genomics, SNPs, transcriptomics, and proteomics, and it's giving you a readout of what happens when the patient's cardiomyocytes are exposed to various concentrations of doxorubicin. Now, in a follow-up study, we showed that the iPS cells can also be used to model Herceptin-induced cardiac dysfunction. In patients with breast cancer, here we recruited 2 patients who did not exhibit any cardiac dysfunction after Herceptin, 2 patients who developed moderate toxicity, and 3 patients who developed severe toxicity after Herceptin with more than a drop in ejection fraction. Now, in contrast to doxorubicin-induced cardiotoxicity, which causes sarcomeric disarray and cell death, as shown in the last slide, Herceptin-induced cardiotoxicity is associated more with metabolic derangement and mitochondrial dysfunction. And importantly, using drug screening, we also showed that metabolic modulation with AMP kinase activators, such as metformin, can be used to rescue the cellular phenotype. Now, besides doxorubicin and Herceptin, other popular chemotherapy drugs include tyrosine kinase inhibitors, or TKIs. Common TKIs that you may have heard of include Gleevec and Sutent, which inhibit VEGFR2 and PDGFRB and can cause heart failure side effect. And so in this study, we generated iPS cells from patients, treated them with most and came out with a cardiac safety index that allows us to predict which TKI is cardiotoxic and which TKI is not. I think this is still very crude at the beginning, but our ultimate goal is to further optimize this cardiac safety index so that we can better predict the safety profiles of chemotherapy drugs before giving them to patients in the future. So as you can see from these examples that I just showed you, we are integrating iPS cell biology with various cellular, molecular, and functional readouts, as well as multi-omics analysis to help us better understand the disease at a systems biology level. I think in the next few years, you will see more and more examples of investigators using large-scale iPS cell population to study various differentiated cell types, such as heart, brain, liver, muscle, and lung, to study expression quantitative trait loci, or EQTL, and also to study genotype-phenotype relationships via co-expression networks. So for the last part of my talk, I want to touch upon how we can use iPS cells to implement the concept of clinical trial in a dish. Here, the concept is that we should be able to recruit patients, generate their differentiated cell type of interest, test which patients respond to drugs and which patients do not, using the iPS cells as surrogate and as a readout, and then go back to perform the actual clinical trial on patients who are positive responders to the drug. And that way you have a higher chance of success rate for your phase 1-2 clinical trials, decrease the cost, and also accelerate the timeline of drug development. So this is an important issue because I think, as many of you know, on average, it takes about 12 to 15 years and $2 billion to develop a new drug. Even worse, many of the drugs that you take today actually do not work at all. And shown here are the top 10 highest sales for drugs in the US. Blue means the drug work, and the red means the drug does not work. So for Abilify, which is used to treat schizophrenia, out of 5 people, the drug only helps 1 person. For Nexium, which is used to treat heartburn, out of 25 people, the drug only helps 1 person. So overall, this is a pretty poor response rate, and this is the reason why the editorial here is titled, Time for 1-Person Clinical Trial. Now, several companies are thinking along the same line. One of them in particular is Takeda Pharmaceutical in Japan. And Takeda has taken advantage of their close partnership with Shinya Yamanaka. This is a April 2015 press release showing Takeda's investment of $268 million in the iPSL technology for developing drugs in the areas of heart care, diabetes, neuropsychiatric diseases, and cancer immunotherapy. What's amazing is that by August 2017, which is about 2.5 years later, the Japanese team already identified the drug hit based on data from the iPSL screening and started on a phase 1 trial. Here, the investigators recruited patients with a rare genetic disease called fibrodysplasia ossificans progressiva, or FOP, also known as Munch-Meyer disease. It is very rare, occurs in about 1 per million people, and has a median life expectancy of 40 years. Patients die when their muscles and tendons are gradually replaced by bone, and it's sometimes called the mummy disease. Because it is so rare, it would not have been possible to do a conventional clinical trial. So here, the Japanese investigators generated iPSL from FOP patients. They screened for 6,800 drugs and identified rapamycin as an effective molecule for preventing bone formation. And they have now started a phase 1 clinical trial by recruiting the patients back. So similarly, in the US, companies and the federal agencies are also thinking about using iPSLs to accelerate the drug development process. This is a study published by several companies and the FDA in which they tested iPSL-derived cardiomyocytes for predicting arrhythmia risk as part of the comprehensive in vitro pro-arrhythmia assay, or CIPA initiative. I think, as you all know, all the drugs that we take need to be tested for cardiac arrhythmia before they get approved by the FDA for human usage. And currently, the platform is testing them on CHO cells, which is Chinese hamster ovarian cells, or HEK cells, which is human embryonic kidney cells that overexpress the hERG potassium channel. It is not ideal because CHO and HEK cells are obviously not cardiomyocytes. And here, the investigators use 1 consensus protocol, 2 human cell lines, 3 continents, 28 blinded drugs, 2 statistical models, and 10 volunteer labs. And importantly, the investigators were able to show an 87% prediction rate, which is very good considering that this is one of the very earliest studies by the FDA CIPA group, and therefore there's going to be significant room for improvement in the future. In our lab, we have also been pushing in that direction as well. This is a paper that we published in 2016 using transcriptome profiling of patient-derived cardiomyocytes to predict individual drug safety and efficacy responses in vitro. Here we tested 2 drugs, rosiglitazone and tacrolimus. We also use CRISPR genome editing to evaluate and confirm the potential mechanisms of action, as well as adverse effects, especially for rosiglitazone, which is a PPAR agonist that was used for diabetic patients in 2000, but then was pulled off the market in 2010 because of increased cardiovascular deaths. Now, as you know, there's a lot of excitement using bioinformatics analysis, big data, and artificial intelligence to predict and risk stratify patient-specific drug responses. In my opinion, I think the problem is that many of the input data are quite noisy, and hence you may end up with a junk in and junk out observation. And I think here, by using human iPSLs at very controlled, defined experimental conditions, you now have a human cellular platform for unbiased validation of your big data or for your AI algorithms. So one of the goals in our lab is to start creating a human encyclopedia of transcriptomic signatures for drug responses using human iPSLs. So previously, I've shown you the transcriptomic profiles of chemotherapy drugs, such as doxorubicin, Herceptin, and tyrosine kinase inhibitors, and also drugs that were withdrawn from the market, such as rosiglitazone. I think for cardiovascular drugs, so we're taking a similar systematic approach, and shown here 2 examples whereby we assessed the effects of 4 different kinds of calcium channel blockers, which are nifedipine, amlodipine, diltiazem, and verapamil, and 4 different kinds of statins, which are atorvastatin, lovastatin, simvastatin, and fluvastatin. So what is interesting to us is that you will see different gene expression patterns among these drugs. And this suggests that even though they belong to the same class of drugs, they may exert different effects within different individuals. And then furthermore, these cellular and molecular effects may be independent from the clinical action of reducing blood pressure for calcium channel blockers or reducing cholesterol for statins. But then I think more work is also needed to confirm these observations here. More recently, we follow-up study on the same large cohort with Lamin cardiomyopathy that I showed you earlier. But this time focusing on vasculopathy, because we noticed that many of these Lamin patients develop early onset hypertension at age 30s and 40s. And so here, instead of studying their cardiomyocyte, we differentiated their iPS cells into endothelial cells. We then performed a whole battery of functional assays and identified KRF2 as potential transcription factor that is responsible for the endothelial dysfunction. And then using a chemical library screen, we found that lovastatin, which is a potent KRF2 agonist, can be used to rescue the endothelial dysfunction. And then we now want to test to see if this is true in actual patients themselves. So the same Lamin patients of whom we tested the iPS cell-derived endothelial cells were then asked to return to our clinic and were treated with lovastatin daily. The clinical data is shown here. We use EndoPAT, which is a machine that can assess flow-mediated dilatation during reactive hyperemia. So normal index is considered 1.67 and above. And as you can see here, Lamin patient number 2 started with a poor index of 0.48 and improved to 1.06 after 6 months of lovastatin treatment. Lamin patient number 3 started with a poor index of 0.61, which improved 0.95 after 6 months of lovastatin treatment, and then 1.5 after 18 months of lovastatin treatment. So this is an example of bedside to bench research whereby we first identify large family pedigree with multiple sudden cardiac deaths due to a terrible genetic mutation in Lamin. It is also an example of a bench to bedside research whereby we screen for already FDA-approved drugs and found that lovastatin can be used to improve their vascular dysfunction. Lastly, I want to touch upon the concept of using iPSL to study ethnic diversity and disease susceptibility. This is an important topic because of the tremendous diversity, ethnic diversity in our country, especially, for example, in states like California. From a health and science perspective, we're interested in understanding why African Americans have much more hypertension, why Hispanics have much more diabetic vasculopathy, and why South Asians, for example, Indians and Pakistanians have more metabolic syndrome. And lastly, I'm also curious in the topic of aldehyde dehydrogenase in East Asians. For those of you who don't know what it is one of the most common genetic polymorphism in the world. This enzyme deficiency is present in 8% of the world population and 36% of East Asians. It causes alcohol flush reaction, which includes flushing, nausea, headaches, and tachycardia. Antabuse or disulfiram works on a similar mechanism and is used to help alcoholics stop drinking because they will feel lousy after drinking. Now, aldehyde dehydrogenase 2 polymorphism involving glutamic acid to lysine switch has also been linked to increased risk of CAD, more severe outcomes after heart attack, higher incidence of hypertension and cancer, as well as increased complications for type 2 diabetes. So now I'm heterozygous for this enzyme deficiency. I know this because my father, he can drink a whole bottle of wine without any problem. And My mother cannot tolerate a single sip. And so my tolerance is about 1/3 of a glass. And if I drink more than that, I will always have facial flushing, tachycardia, and headache. So it's a good enzyme to be missing because it's hard for me to become an alcoholic. And I also don't need to exercise because I can just have a glass of wine before I go to bed. And that's my exercise for the day. But, you know, joking aside, I think this enzyme deficiency has been well documented to be a risk factor for CAD and myocardial infarction in most East Asians. And here you can see in Japanese men, Korean men, and Han Chinese. So when I saw this data, I had to prioritize and ask my postdocs to work on a stat. And so here we recruited 10 Stanford undergrads who are of East Asian descent. 5 of them are wild type and 5 of them are heterozygous, which means they'll have similar symptoms like I do: headache, flushing, tachycardia. And if you're homozygous mutant, which is about 1%, the symptoms get even worse. And here we model myocardial infarction by exposing the cells to hypoxia. And as you can see here, the wild type cardiomyocytes tolerated hypoxia quite well with minimal apoptosis, whereas the heterozygous mutant cardiomyocytes have significant apoptosis with hypoxia. And I think overall, we believe that the enzyme is not only involved in detoxifying alcohol, but it's also involved in getting rid of reactive oxygen species, or ROS. And so that after a heart attack, there'll be tons of ROS that get activated. If you're missing this enzyme, you have harder time getting rid of the ROS, and hence leading to more cell death and perhaps explaining for the worst clinical outcome that is observed in epidemiology studies. I think this is just one example of using race-specific iPS cells to study ethnic differences. And I think undoubtedly you will find more and more examples of investigators using iPS cells from different ethnicities to study various biological questions in the next few years. So finally, I just want to highlight the Stanford Cardiovascular Institute iPSL Biobank. So far, we've generated cells from more than 1,000 patients with different cardiovascular history, ethnicity, and sex, in collaboration with the NHLBI Top Met program. We're in the process of finishing DNA sequencing on all these lines. We also use FarmGK database to understand how human genetic variation impacts drug response phenotype. We're also working with the NIH on biobanking and with FDA on drug safety testing. Lastly, we have established a very robust sharing resource plan with many investigators, so that if you're interested in some of these lines, please feel free to contact me and we'll be happy to send you these cells. So in summary, I hope I have shown you the various applications of iPS cells for disease modeling, for cardio-oncology, for clinical trial in the dish, for disease modeling. I think in addition, genome editing is equally important to this equation, and this is as witnessed by the recent Nobel Prize in Chemistry awarded to CRISPR inventors Jennifer Doudna and Emmanuel Charpentier. And I think together with patient population, clinical phenotype, and genomics, they constitute what I believe some examples of the initial steps toward precision cardiovascular medicine. And I think, in my opinion, perhaps the most important advantage is that they come from human sources, and hence they can be used to recapitulate the diversity of human genetics that is otherwise not possible with conventional animal models. And that's the main advantage of why it's a powerful tool for biomedical research. So this concludes my talk, and I want to acknowledge my postdocs, my graduate students, cardiology fellows, and instructors, and their funding support. And I also want to acknowledge some of my former trainees whose work I highlighted here, and also my collaborators and my funding support here. And I think, Ryan, I'm listed as co-investigator and co-PI for many other people's grant to help out with their grant application. So I actually don't have that many grants, as you pointed out. But thank you so much for the invitation, and I'll be happy to answer any questions. Yeah.
Joseph, that was just an amazing tour de force. Thank you so much for spending our time to speak at our research day. We really appreciate that. So I have two questions. Are there any scenarios where iPSCs don't work? In other words, they have proven to be perhaps somewhat different from either the clinical syndrome when you have applied it in the ways in the way that you have very nicely discussed for largely monogenic disorders.
Yeah. So that's a very good question, Dale. So I think most of us go after monogenic disorders because they're easier to model. I think if you're polygenic, it gets to it gets more difficult. And I think if you have socioeconomic environmental factors, it almost becomes not possible to model that, right? So the ideal example I try to give is that hypothetically I have an identical twin, and my identical twin is a perfect athlete and exercise all the time, whereas me, I got a divorce and I'm all depressed and I gain 100 pounds and my BMI is 35 and get a heart attack. You generate the iPSC cardiomyocytes or iPSC endothelial cells. You're trying to understand why is it that Joe Wu got a heart attack? Why is it Joe Wu got diabetes? But in that case, a lot of that is due to socioeconomic and environmental factors. And your cell will look exactly the same as your identical twin, who's actually perfectly healthy. And if you were to generate iPS cells from the identical twin, your question at that time will be different. Your question will be asking, what are the cells that belong to a healthy, vibrant individual? So I think as clinicians, we have a better insight in terms of what's possible, what's not possible. For the environmental factors that you cannot recapitulate, you can actually use the cells to model that. So for example, I didn't show the data, but we have projects in which we take iPS cell derived endothelial cells to model exposure to nicotine for smoking, to model exposure to some of the components of the e-cigarettes for vaping. And in the case of other examples, let's say, for example, diabetes, sometimes it's harder because you don't know whether the diabetes is due to genetic component or due to habituatory components. So we've recruited type 2 diabetic patients, but we typically go after the ones that are not obese and large family because we're betting that those patients would tend to have more genetic components. And then we would expose their cells to the patient's own serum. So, getting back to your question, there are examples in which it doesn't work because of the question you're framing cannot be recapitulated. There are also examples in which it doesn't work, in which the differences in your iPS cell reprogramming differentiations this much, but then the biological difference is this much. And because your technical variability is too big compared to biological variability, you cannot see a difference. But in that case, I will say that over time, that's going to correct itself as more and more people work on the iPS cells and the differentiation technique becomes better, more robust, more reliable. The biological difference, the technical difference will become smaller, and hopefully we'll be able to recapitulate the biological difference that's bigger than the technical difference. Yeah. So a long-winded answer to your question. Yeah.
That's great.
I have a question, if I may ask. I'm Mary Wilson, and I also really enjoyed your talk. Does one, in order to use the iPS cells, does one need to know the genetic admixture of your population first?
Can you repeat that question again? I'm sorry, Mary. Yeah.
Since a lot of the changes are, as you pointed out, sometimes determined by genetic background, which can be specific for different populations. Do you need to know the admixture, the genetic admixture of your population before doing the experiments? Because oftentimes you don't know,
Yeah. So that's a perfect question. And so I think, times people would ask us for cell lines and say, can you give us some control healthy cell lines? And I always have to paraphrase it by saying, there's no such thing as control healthy cell line, meaning that if you take my iPSL when I'm 20 years old, yeah, I send it to you as control healthy. But then, when I'm 80 years old, I may have prostate cancer, diabetes, multiple heart attack. You would have coded that cell line as prostate cancer. So what we could tell the investigators is that we're sending you, to the best of our knowledge, genetic versus non-genetic. And we restrict it by saying, we don't think these lines that have genetic causes of heart disease, meaning that a lot of times the cells have been screened either clinically in our clinic or we've undergone, most of our cells that we do SNP karyotyping. And some of the cells, as I mentioned, actually most of the cells have undergone DNA sequencing by the NHLBI top med. We're waiting for their results to come back. And so once they come back, we will have a better idea of what type of genetic background they have. But right now, I think it just really depends on your question. If you want to be perfectly sure that a particular disease that you're working on, and the reviewers are asking that, you may just as well undergo a DNA microarray or undergo exome sequencing just to confirm that the line doesn't have that particular mutation. I think the difference here is that, why we like the iPSL more than human ESL. Both of them you can sequence to understand the mutations of the variants. But then as I pointed out, a lot of times when you sequence, you get tons of variants. You don't know how to interpret that. Yeah. Human ESL, I cannot interpret that because I don't know who the patient is. Whereas the PSL, I also have the patient and I can go back to the clinical database and say, okay, I did the sequencing and it says has some variants that causes hypertrophic cardiomyopathy. But clinically, the patient is already 70 years old and I just don't see it. So that's why it gives you more information in terms of the iPSL compared to the human ESL.
Okay, thank you.
Now, have a question? Ajit, wonderful talk, thank you. And what about the gender difference? We normally see in animal model, but in your IPS system, can you still tell the distinguish the gender difference?
I think your mic is kind of muffled, so I think your mic is kind of muffled, so I didn't catch the question. Yeah.
Oh, okay. I mean, the gender difference from male and female.
Oh, the male versus female. So we haven't looked into that. We've asked that question, but we haven't exactly looked into that. I think it becomes more. I think you'll probably see some difference, but you also have to control for the fact that in human body, males and females have much big differences in terms of testosterone, estrogen, whereas in cell lines, the culturing condition, for example, some of the media already has some estradiol. So if you're gonna try to do this type of research, you gotta make sure that you do a charcoal depletion of your media to make sure there's no estradiol. You control your level of the estrogen and then also control your level of testosterone. I think, for example, for many of the hypertrophic diseases, they manifest during the puberty phase. And so in the past, we've always wondered, what is it about the hormones that causes the disease to manifest more? And these are some of the questions that you should be able to model in your iPSL platform, but you just have to make sure to control for the inherent levels of estradiol in your cell culture media.
So,
hey Barry, yeah.
So wonderful talk. Amplifying a little bit on your answer to Dale's question, how reproducible are IPS cell findings nowadays? Labs, each lab differentiates cells a little bit differently. How often are there? Do you think that mucks up the data and/or that that there's that the process of doing the experiments differently sort of makes a real difference?
I think there's a big component to that because each lab does things differently. And I think the other flip side is that as more and more people work on it, I think naturally they'll gravitate towards some kind of commonly used protocol or commonly used cell line. And this is part of the reason why we have an open sharing policy. For example, I was mentioning to somebody earlier that over the past 4 years, we sent out 2,500 vials of these iPS cells. And the goal is that, if you want to study these cells, if you want to study long QT1, long QT2, yeah, we have these cells. We're happy to send it to you. You can study it. We share our differentiation protocol and see what you get and compare to the existing data in the literature, and then also compare to what other people, what all labs have done. So just like, I don't think this is just a iPS cell field. I think this is a science in general that there's a lot of variability. And I think one way to address this is, open sharing policy, and more people, as more people work on it, I think it's higher chance that they're going to come to common theme, common.
How close do you think we are to sort of really standardized protocols for creation and differentiation?
It's difficult, right? So I think for the SHIPA initiative, that we are part of the SHIPA initiative, we standardize the protocol for that. And all the labs decide to have the temperature, the amount of the media, everything is all standardized. And so that there's no variation. And this is very important for the FDA. And you'll see that the publications from the SHIPA initiative, in which everything is all standardized, you'll see that Stanford's data looks very similar to Hopkins data, looks very similar to Amgen's data, once it becomes standardized. But for academic research, it's difficult. It's difficult for us to tell how this standardized. Yeah.
Thank you.
Okay, I think due to the problems posed with using Zooms, we have to end this session and get moving on with our next session to allow those breakout rooms to open up. On behalf of the organizers and everyone who attended this keynote, I just want to thank you, Joe, for an outstanding talk. One that I think appealed very nicely to clinicians and basic scientists here at Iowa, and really did demonstrate the power of these stem cell-based methodologies and their place in medicine across quite a diverse set of biomedical paradigms. So it's a great talk. Thank you very much.
Thank you, Ryan. Thank you for the invitation. Good to see everybody. And as I mentioned, we're happy to share all these cell lines, and just send me an email, and that's how science advances.
Awesome. Thank you, Joe.
Okay. Bye.